So the 12-hour is a 3.4-fold increase from the 24 hours when we're talking about CH007, and a 4.6-fold increase when we're talking about KB1. So that's a pretty significant increase in particles that we're harvesting from just simply the 12 versus the 24 hours. So now we'll go into the chemical analysis portion. We take our three fractions. We have our lyophilized cell pellet and MV pellet, as well as the MV-free supernatant. And we extract these with various parameters, resulting in our chemical extracts, and then we subject these to two different mass spec techniques. We're using an LTQ to look at molecular networking and to de-replicate the structures of any potential prodiginines, using MZ-3 and GMPX, and then we're subjecting these fractions also to triple quadruple LC-MS to quantify the levels of prodigiosin using a standard that we developed with a reference of prodigiosin, pure prodigiosin. So going forward, pure rubra extracts of the MVs do contain prodiginines, which is awesome. We did find all of them in the CH007 and all but one in the KB1, but here are their ticks, as you can see to the right, and here's for CH007, those splitting patterns that we matched up in a literature review. So we do validate that we have our prodiginines, namely cycloprodigiosin, which has been correlated to the larval settlement, as well as in the KB1. All right, so going forward with our quantification, here's the big hurrah. We have our MVs, and the total prodigiosin that we are calculating using particles per mil equivalents. We have 27 micrograms for CH007 24 hours, and then 1,317 micrograms for our 12 hours. So if you remember, the differences between the 24 and 12-hour MVs total harvested, those were only a 3 or 4-fold increase. But looking at this, the total prodigiosin harvested is actually more like a 40-fold increase. So it's not just that the 12-hour MVs or the 12-hour cultures are producing more MVs, they're also packaging more prodigiosin within their membrane vesicles. And similarly in the KB1. The cell pellets and CH007 24 hours is representative of all cell pellet extracts, but the total prodigiosin is a very, very high number, well over 7 billion. And we have the MV-free supernatant with less than 0.1 micrograms. So this is significant because these two fractions contain our prodigiosin and the supernatant doesn't. And this would make sense because prodigiosin being a tripyrrole alkaloid with an unsaturated chain of various lengths would make it a very lipophilic molecule. And if we're talking about chemical communication in the marine environment, in an aqueous solution, the lipophilic compounds would have to be packaged in some sort of drug delivery mechanism, and that's what we're suggesting that these membrane vesicles play the role as. So now we did some bioactivity testing, and this is some preliminary data. It definitely needs to be optimized, keep that in mind. But essentially we're comparing the 12-hour MVs and the 24-hour MVs with their bactericidal effects against B. angularum, which is essentially a marine pathogen that infects salmon. But it does have activity, and these vesicles per mil are very similar. However, there's a little less, obviously, in the 12-hour MVs, potentially because they're packaging more prodigiosin. And prodigiosin itself we used as a control using that reference standard that we had. And we know that the MVC is somewhere in between 5 micrograms per mil to 0.05 micrograms per mil. And we also know that our solvents that we're dissolving these in have no activity because of our controls. And this bactericidal effect is sustained up to 8 hours, and if not, increases over that span of time. So more to come on that. So I just want to remind us of our hypothesis as well as our questions. So do CCA-associated bacteria produce membrane vesicles? Yes, they do. P. rubra is a prolific producer of membrane vesicles. Do they mediate chemical communication? Not sure yet. That will be coming soon. And if so, what cargoes can be found inside these MVs? We found a number of prodiginine analogs, namely prodigiosin, and there we go. We labeled them as prodiginines. So conclusions and future directions. P. rubra strains produce MVs containing these prodiginine antibiotics. We are not suggesting that these are the only chemicals responsible for their bioactivity. We will be subjecting the membrane vesicle extracts to metabolomics as well as proteomic studies to further validate and gain more data. And they do have rapid antibacterial properties against the marine pathogen V. anguillarum. Future directions include whether or not they induce coral larval settlement, which those experiments have actually been set up this week and will be going on during the next, so I'm very excited to get that data back. And the MVs also, whether or not they have broad spectrum antibacterial effects against a wide variety of marine pathogens. Being that we're in a probiotics facing marine pathogen lab, we have a number of different strains that we'd like to test it against, in particular Vibrio coralliticus, which has been associated with infectious disease within the coral ecosystem. So some acknowledgements. I'd like to thank the URI team, the Rally Lab in particular. Lolade was a big help with any LCMS MS data gathered. RV and Elise were handling most of the antibacterial assays. Roger Williams University, they're taking care of all of the larval settlement assays, and I'm very excited to see what they come up with next week. And the URI Probiotics group has always been a big help when designing experiments and providing advice. And Matt Burton, who taught me most of what I know about mass spec. So much thanks to them. Be sure to check out RV's poster. She's going to be going tomorrow in the second poster session, and she'll be talking about membrane vesicles from a couple other strains of Pseudoalteromonas that we have. And I'd also like to thank ASP and the organizing committee for giving me this time to talk to you all and hear my research. Thank you so much for listening, and I'll now take questions.
Another fantastic talk. How was that for a tour of a bunch of different ecosystems? Thank you all of you who stayed. And I see people grabbing for the microphone, and Leslie Ann.
Hi, Leslie Ann Giddings from Smith College. Great talk, Maggie. Just a quick question about the impact of the light sensitivities of some of the prodiginines on the MVs and their bactericidal activities in the environment. Do you have any ideas of what might happen?
Yes, that's actually a great question. We found really early on when we were isolating these prodiginines that if we left the extracts in the fridge in just a glass vial, sometimes they would decrease in their quantity. So we suggest that this is probably light degradation, very much like what the Shup Lab group discovered. So we've been keeping them in those dark vials ever since because they are subject to degradation. Great question.
Thank you so much for a great presentation, Krzysztof from the University of Alabama. So actually, you are hypothesizing that those vesicles might be involved in attracting the larvae settlement. So in a real world scenario, there would be some probably a gradient of those, right? Like that gradient concentration high, close to where they're released, and decreasing further from that release side. So I was wondering if there's some sort of like chemotaxis happening. So are there any specific receptors, protein molecules that might be playing a role that are on the surface of those vesicles that will be the one that the larvae might be looking for? They have specific receptors to basically find, and then let's give them the signal, okay, we're going in a good direction.
That's a great question. So membrane vesicles could be uptake by a number of cells via endocytosis, but receptor-mediated endocytosis definitely. And we have not looked into the proteomics, which probably would indicate more if there were receptors present on the outer membrane vesicles. However, the prodiginines themselves we suspect to be lodged within the membrane vesicle because of their long unsaturated chain. So potentially the prodigiosin itself is acting as that recruitment molecule.
Awesome, thank you.
Thank you.
So I think we are starting to run into the poster session, but I think the speakers are mostly still here, and so grab them if you have more questions for them or one-on-ones. Let's thank them one more time.